Can everyone hear me okay? Yeah, just put your hand up if I go quiet. So at this conference and also for many years we hear a lot of speculation and discussion about the evolution of language, and many of the discussions centre on the roles of genes and on kind of genetics of language, if you like. And of course we've heard also of the issues of importance of other factors like culture. I'm not really going to deal with that today. But what I want to really try and the take-home message of my talk is that if we're going to make any kind of sensible discussions about the genetics of language, if there could be such a thing, this needs to be constrained by an understanding of the language of genetics. So I want to kind of start by debunking a particular myth, which I call the abstract gene myth. And it's this idea that, of course, we're hearing more and more about how different genes affect different aspects of behaviour, and this tends to lead people to think that there are these direct lines between genes and something such as speech and language, something as complex as speech and language. So there's this kind of view that genes are these abstract entities that sit around in our bodies with a mysterious ability to directly control our behaviour or our cognitive skills. And unfortunately, this is just not true. Genes really don't behave like this. This direct line doesn't exist. So we need to replace this view if we're going to have discussions on the same page with something a bit more realistic. And here's another view of the gene, which perhaps is not even helpful either, because this is all the strings of A's, T's, G's and C's that I spend much of my day staring at and getting gradually more and more mad. But luckily for geneticists, we know a lot about how to decode some of this information. And particularly, there's one type of gene that we understand very well, better than others, and that's the type of gene that codes for proteins. And that already tells us a lot. And the way this works is, and I'm illustrating this in very simplified terms and very quickly, just to get everyone up to speed. This is my very simplistic view of the double helix, the double-stranded DNA. This gets copied into a copy of one strand, which is a messenger RNA, a single-stranded copy. And it basically has the same information that's coded in the DNA, in this strand of the DNA. It's just a copy of it, except that instead of A's, G's, C's and T's, there are A's, G's, C's and U's. This doesn't matter. So the main point then is that this acts as a template for a code that's a triplet letter code, and it involves 20 different possible amino acids, and each of these different 64 possible combinations of 3 letters codes for a certain specific amino acid. So this is the ultimate, or not the ultimate, this is a very nice combinatorial system which gives you kind of discrete infinity, because you have ultimately, you can assemble with this code a limitless number of sequences of amino acids of different lengths and with all these different combinations. And the amino acids, each of the amino acids differs in its properties. So you can basically go from a single letter nucleotide code, a linear code, to a linear code in amino acids. Why is this important? Because the linear code of the amino acids determines the shape, the way that the protein folds up. And why is that important? Because the shapes of proteins are what determines what a protein does. The function is specified by the shape. So the function of this protein is specified by its shape. Its shape is primarily specified by its amino acid sequence, and its amino acid sequence is specified by the RNA sequence, which is basically the DNA sequence. So this is, as I say, this is a very fast-track kind of tour through the protein coding genome, and there's other aspects that I haven't got time to go into. But this already constrains how genes can behave, right? Because these protein coding genes, as we've heard already a couple of times, these don't actually code for behaviours, they code for proteins. In the human genome, you have now, it's thought about 20,000 or so protein coding genes. Now, there's an extra added complexity that is important to mention, and that is that in between these genes, there's these big gaps in the genome. And in many cases, we're beginning to discover what these things do. But one of the key things that we know is that in front of every little recipe, every little protein coding gene, there's something called a promoter that determines whether the gene's going to be switched on or off. Because, of course, you have 20,000 different genes, but you don't want them all switched on at once in every cell in your body. This is what gives your cells different functions, and also it allows us to respond to an environment and also to have internal responses. The genes need to be dynamically being switched on and off during development and actually in mature animals as well. So the genome doesn't just contain this information, the sets of proteins that are needed to build the building blocks of cells, but it also contains the organizational information telling the genes when and where to be turned on and off. And this consists of two factors. One is the regulatory sequences in the genome itself, such as the promoters, but there are other ones. And then we have proteins that actually stick to these things. And so you might have a protein that sticks to here to switch on this gene. Now, these proteins are themselves coded by genes. So you immediately see that you have these very complex networks building up. And so the biology of an organism results from a complex series of these switching events and developmental events, moderated by lots of other important things like environmental influences and stochastic influences. So that's one sort of reason why you don't get these direct lines. It's a little bit worse than that as well, without wanting to discourage you more from these kind of things. So we know that the DNA, as I said, we know that it codes for protein, and we know that those proteins then act in cells and they're switched on and off in dynamic ways. But when it comes to the nervous system, we've got an even more complex thing to worry about, which is that the nervous system is composed of a really diverse mixture of different neuronal subpopulations wired up in really complicated ways, in interesting patterns. So not only do you have diversity of cell type, but you also have this wiring dynamic. And the wiring, these neural circuits, then have their own properties that determine how they're going to respond to the brain. So on top of the genetic pathways, you've got this diversity at this level and at the brain level as well. And of course, I haven't gone into these other levels that we've heard about of cultural transmission that can be really important for speech and language as well. But what I really want to emphasize here is that you've got, in order to understand how genes relate to something like behavior, you've got to be thinking about all these different levels, which is quite a trick to do if you can do it. Any kind of discussions about what genes are going to be doing in the context of evolution of language or function of language has to really think about these constraints. These are constraints, if you like. Now, you might be now thinking, well, this really makes it hard for us to even think about making links between DNA and speech and language, so maybe, Simon, you should just give up and go home right now. And actually, it's even harder than that, because, of course, geneticists who are interested in other traits have got all sorts of other organisms that they can rely on to study their traits of interest. So if you have a phenotype that goes across the animal kingdom, you can look at it in other species. And here, we're limited, at least if we really want to look at complex spoken language, to this little one twig on the tree of life. So what do we do about that? Well, as it happens, it's not all doom and gloom. So here's the good news. Variety is the spice of life. So although we know that the whole genome sequence was determined, and everybody carries a copy of this, unless you're dead, and even if you're dead, actually. But I'm going off the point. But everybody has a copy of this whole genome and has the same sets of protein coding regions and regulatory regions and genes. But we all carry these mutations, these variants, these different versions of these genes. And in some cases, we're missing genes and we're carrying extra copies and things like that. One of the big interesting findings of the recent years has been that this diversity amongst us is really high at the genomic level. And in some cases, we carry around these changes and they have no impact on our development, on our biology, and they're just sort of masked in the genome and they're passed along to different generations. But in some cases, there are changes that will change the code of a protein or change the way that the protein is switched on. And in the most dramatic way, it might just sort of take one of these amino acid triplets, and instead of it, there's a type of code that actually says stop at this point in the protein, because otherwise your protein would go on forever. So there's certain triplets which code for stop signals in the protein. So if you had a mutation to a single letter which inserted a stop in the middle of your protein, then you're not going to make a full-length protein, and it might not work properly, and you might have a severe disease, and you might have all sorts of different problems. And the other thing you could have would be other changes that actually affect the particular amino acid that's in the protein and changes the shape of it. So these kind of variants, these kind of missense changes, can also have dramatic effects on protein function because they can really seriously disturb the shape of that protein, and it won't be able to do its job in the cell, whether it's an enzyme that's catalyzing a reaction or it's a channel that helps things get into the cell, or all these different diverse protein functions that proteins can have. So, but you also have some sort of quieter changes that have only subtle effects on how a protein works or on whether it's being switched on or off. So there's a whole range of these changes that are sitting around in all our genomes. And in some cases they are causing disorders, in some cases they have clear causal pathways, in some cases they predispose us to certain disorders. And we can use that diversity as geneticists. We can study the diversity in human populations, or we can study disease, and we can try and relate that to the underlying genetics, and even without an animal model, what we're interested in, we can find the cause. So this is what we kind of where we tried to go some years ago with genetics involved in speech and language, and in fact in speech and language disorders. And this has proved to be one of the easiest ways of getting into these kind of pathways. So a subset of children, as you know, most children have this amazing ability to just sort of very rapidly, without a kind of explicit instruction, to acquire very complex spoken language with fairly basic kind of stimuli. And a subset of children grow up in very language-rich environments but just don't develop normal skills. Sometimes there's an obvious reason for this. So sometimes they've got a clear physical problem, or they can't hear properly, or they have general mental retardation, intellectual disability. But often these cases of unexplained language impairments have no overt cause, and there's no obvious kind of neurological cause either. And so you end up with a kind of mixed bag of kids with language impairments, right, where it's kind of an exclusionary thing of saying, well, they should be learning to speak properly, but they aren't, and we don't know why, we don't know what the cause is. And right when the first of these cases were being identified in the literature, I mean, I think over 100 years ago, it was already noticed that developmental language disorders tend to cluster in families, which is, of course, a kind of clue to a shared family effect, which could be genetics, but maybe something else. And then there have been loads and loads of twin studies of disorders related to language development, many done by Dorothy Bishop and by Bruce Tomlin, and others. And these twin studies suggest that there is a high heritability. So there's a big role for genetic factors in predisposing a kid to whether they're going to have a language problem or not. But the problem here is that these studies were going on for some years, but they don't tell you where in the genome, they don't give you any clue, in fact, where in the genome these things lie. So a lot of what we've been doing over the past couple of decades is to take families and try and track down the genes that are involved. And this is still a tricky task because there's no simple genetic basis, there's no single gene that acts across all these families, there's probably multiple different genes, and they might have very subtle effects, and they might be interacting with each other. So in fact, where we've made the most progress was with this very unusual case that many people have heard about, of this family that jumped out at any geneticist who looked at them, because they showed this really, really striking inheritance pattern. 3 generations of where half of the members are affected with a severe speech and language disorder and half of them are unaffected. And this family were first identified in the... Oh, sorry, I just cut out something. This family were first identified in the late 80s, early 90s by a team at the Institute of Child Health, Faranavarga Kadem, Jane Hurst and colleagues. And they were found because a bunch of the kids were at the same special language unit, and then they traced out the whole of this family tree and found this amazing inheritance pattern. Males and females are affected equally, and there's about half of the people, particularly if you look at this generation, you can see half of the people are affected and half are unaffected. And what I'm going to do now is I'm going to play you, hopefully, if it works, some sound from some years ago when the kids from this third generation were being interviewed, and you can see what you can make of what they're saying.
They are sure. It's the fun. They are 6. The mom is taller. That is how I live in England and understand. Not the cow. I live in England and I like them.
Okay, now each of those children was saying their name, where they lived, and how old they were. And I'm going to play it to you again so you can see that that's what was happening. But you can hear that there was something wrong with, or something strikingly abnormal about how they were speaking. Hold on a minute.
They are sure. It's the fun. They are 6. The mom is taller. That is how I live in England and understand. Not a cow. I live in England and I like them.
And you can hear that the eldest kid was who's had the most speech therapy is most intelligible. But basically, even in adulthood, they have problems with speech articulation. And the kind of problems that they have are they make errors that are inconsistent and that are affected by the complexity of the utterance. And I can show you something here, which is from a paper by Kate Watkins when she was working with Farron and Barbara Cadden in London, where they've looked at the kids in the family. And what you see on the top line here is the unaffected members of the family. What you see here is the affected members of the family, and these are adults with Broca's aphasia. And these are tasks where they have to repeat simple or complex nonsense words, pronounceable nonsense words such as contrampanist and perplistronk that I practice every morning in the mirror so that I can do these talks. But for them, they're new words, or they were when they did these tasks. And you can see that there's an increasing deficit with the number of syllables in the word and also the complexity of the vocalization. These are non-words with consonant clusters. So this is classically known in the States as childhood apraxia of speech, which is a kind of quite a rare type of speech disorder. And it's also called developmental verbal dyspraxia in the UK. And it seems to be about difficulties in learning and production of these sequences of mouth movements, which affects orofacial mouth movements generally and also impairs speech development. So this has been argued to be perhaps the core deficit in this disorder in the family. But it's really important to emphasize that there's other things going on. So it looks like simple inheritance, but there's a complex phenotype. So they have deficits in a wide range of language-related skills. This is not surprising that they have problems with spoken language because they have problems with speaking, but they also have problems in the written domain. So if you have tests of verbal fluency, for example, in written domain, they show reduced verbal fluency. They have impairment that's not just in expression of language, but in also receptive language. And much fuss was made about this at one point in time, some years ago, but they also have problems with comprehension and production of grammar. And in fact, some linguistic explanations were very specific about one aspect of morphosyntax. This is clearly not the case. They actually have a broad-ranging language impairment that affects pretty much all aspects of language development, but seems to be more profound in the expressive domain and in particular in relation to speech sequencing. I should say that in anticipation of some later discussion, that they do not have general motor dyspraxia like general deficits with movement with their limbs. It seems that they have quite severe problems with orofacial coordination, but less so with other tasks. So, another question is whether this is an intellectual disability disorder, and it's not. But the KE family are not the brightest family in the world. There are some kids with low non-verbal IQ in the family. But it's important to notice that they don't segregate very tightly with the disorder, so you have some unaffected individuals who have low non-verbal IQ, you have some affected individuals who have normal non-verbal IQ despite having speech and language problems. And something that's been noted in the literature in studying all other kids with specific language impairment and language-related disorders is that even when they start out with a non-verbal IQ that matches their peers, often kids with language impairment, as they develop, as they get older, they don't keep up with their peers for various reasons that we could discuss later. So this is in line with what's seen in other kids with language impairments. And if you look, Kate Watkins did this, if you look across different kinds of aspects of verbal and non-verbal cognition, you see that the deficits in verbal cognition are much more severe and wide-ranging. So there's clearly something disproportionate going on in relation to speech and language development, even if it's not sort of specific. So I was very, very lucky to have the opportunity to work on the DNA of this family when I was a postdoc in Tony Monaco's lab in the late 90s. And working with a PhD student, Cecilia Lai, we tracked down the cause to a mutation in this gene called FOXP2, which was a novel gene at the time. And the reason it was called FOXP2 was because this is a picture of the protein that it makes. Each protein has a sort of what's called an N-terminus, which is the start point, if you like, and a C-terminus, which is the end. And then this is about 715 amino acids long if I just stretched out the whole protein instead of letting it fold up properly. And you don't need to worry about this as a big stretch of glutamine residues, and this is another domain that's interesting. And here's another domain. But this is what really gives FOXP2 its name. It's called a fork-head box domain. And the reason it's called FOXP2 is because there's lots of other genes in this big family of fork-head box genes that all do similar things, which I'll tell you about in a minute, at the cellular level. And this happened to be in the P branch. It was the second FOX gene. So a nomenclature committee, a committee of people who decide about what genes are called, decided for us that this was called FOXP2. So we're stuck with that name, for better or worse. And the key thing about this fork-head box domain is that it immediately gave us clues to what the protein was doing to its function. And here is a schematic that shows you the shape of the protein, what it looks like. And what you can see, this is just for this one section of it, this 80 amino acid section called the fork-head box domain, which then another group have managed to crystallize. And then you can observe using X-ray crystallography, you can observe the actual structure of it. And it forms these three helices here, helix 1, helix 2, and helix 3. And this is showing it sticking to DNA. What this domain likes to do is stick to genetic material, stick to DNA. So FOXP2 the gene makes a protein that likes to stick to the DNA of other genes. And this third helix is a really key part of the domain because it gets buried deep into the major groove of this purple DNA that you can see here. And what we found in the KE family was a very simple genetic change. So just a change of a single letter of DNA. I don't know how well you can see that here, but I'm going to show you on here because it's a bit closer. But at this particular point in FOXP2, everybody in this room, I think, and most people in the world carry a G. In the KE family, somewhere in the grandma or in one of her parents, there was a mutation of one copy of the gene so that it had an A instead of a G. So the KE family affected members all carry this mutation. And they carry everybody has one copy of a gene from the mum and one copy from the dad. So you have two copies of the gene. In this case, just one copy is damaged. So they have one normal copy, and then they have one copy that has this A instead of a G. What does that do to the protein? Well, we can immediately say, because, in fact, it changes the code of the protein. And this is an arginine. R stands for arginine, a particular type of residue, 553 amino acids from the start of the protein. And what happens here is that this gets mutated because of this change. The code then becomes a histidine. And what we've been able to show is that if this protein carries a histidine instead of an arginine at this key point of the recognition helix, it can't stick to the DNA that it normally likes to stick to. Now, in that this is often neglected, but in that first paper in 2001 that we published in Nature, we had another case of a child unrelated to the KE family who had a very similar kind of profile of speech and language problems, and he was unusual because, in comparison to the KE family, because he was the only case in his family. He just was a de novo mutation, and it was a different type of mutation from this one, which is a sort of missense point mutation. This was a big breakage in his chromosome. So part of this gene lies on chromosome 7, and part of his chromosome 7 had broken and become attached to part of chromosome 5. And the important thing about it is that he has all the genes, but what's happened is, at this particular point of FOXP2, it's broken in half. So he has a breakage here, and then it's stuck to chromosome 5 on both sides. So one copy of FOXP2 is intact, one copy doesn't work, similar to the KE family. And something else that's often sort of disregarded is that there are other mutations around as well. So here is an example of another mutation that we found in a small family. So we screened about 50 kids with similar problems, this developmental verbal dyspraxia, this childhood apraxia of speech. We screened 50 kids across the whole of FOXP2, and we found only 3 mutations in those 50 kids. So it's very, very rare to have mutations of FOXP2. But this is the most interesting of the mutations because, remember I said that mutations can insert these stop signals. And this is exactly what this mutation does. It inserts a stop signal in this kid's FOXP2 gene, which basically means that the protein stops at this point. And so you're missing the DNA binding domain and all sorts of other things that you need. So, interestingly, this kid, when we studied him, he didn't come from a big family, like a KE family, but he had a younger sister who was, it turns out, was developing the same problems that he had, so developmental verbal dyspraxia, and a mum with a history of speech and language problems. And each of them carried this change, and again, they were heterozygous, one damaged copy, one normal copy. And I should say that since then, there are now maybe a dozen, 20 other cases of people with damage to FOXP2 and with speech and language problems, sometimes in the background of other deficits. And the reason for that is because most of these other cases are deletions that don't just delete FOXP2, but delete other genes as well, neighbouring them. And there are also a couple of other translocation cases that have been found as well, where there's these chromosome breakage cases. So they're not as clean as these cases where you just have, like in the AD family or in the KE family, just a single letter being changed in the gene. But all the cases that we've seen so far point to the idea that if you have a reduced dosage, if you're only making half of the normal FOXP2 protein, then you have problems with developing speech and language for whatever reason. Now, the other thing that we know about FOXP2 that's really important is that FOXP2 is a transcription factor. So I already explained a bit about this to you. So to return to this very simplistic double helix picture, this is, if you imagine this is the FOXP2 gene, something comes along and switches on FOXP2, and the gene gets turned into protein. And then the FOXP2 protein, which is made outside of the nucleus, then goes back into the nucleus where other genes are, where the DNA is, and it finds so-called downstream targets. And it also, to do this, it interacts with other proteins, which aren't shown on here. And what it could do as a transcription factor is either it can stick to the promoter of a gene or a regulatory sequence and switch it on. So target gene X is here being activated. Or another route of action could be for this gene to stick to the front of the gene, the promoter, or another part of it, and silence it, switch it off, repress it. So what we think that FOXP2 does most of the time is to repress genes, is to silence genes. But what I've shown you is a simplification here, because actually FOXP2 doesn't have a kind of all or nothing effect, as I've shown here, but it's like a genetic dimmer switch. It tunes down expression. It's sort of, it's much more subtle in its actions than just activating or repressing. But it could be a hub in a network of different genes. What we've been able to show, and I don't have time to go into all of this, but we can grow human neuron-like cells in the lab. We can culture them in the lab, and then we can stick in different versions of FOXP2, the versions that we found that are mutated in the disorder, and we can ask how they function in the cell. And these are just some examples of pretty pictures showing you FOXP2, normal FOXP2 in green here, sitting in the nucleus of a cell. And for example, this mutant FOXP2 never ever gets into the nucleus. It can't get into the nucleus. It stays, and it doesn't work properly. So we have lots of work we've done on that. Was the first paper we did on this, but there's lots of work we've done on that, which pretty clearly shows that when you have these mutations, they're messing with the function of the gene. So what I really want to emphasize from this is what we know then is that if you have damage to one copy of FOXP2, it messes with your speech and language abilities. This is not the same as us saying that this is the gene for language, and in fact we've never said this. So versions of FOXP2, and this was already apparent in the first paper in fact, versions of FOXP2 are found in a very similar form in other species that lack speech and language, mice, birds, maybe we'll talk about those later. I'm not quite sure what that species is, actually. But so it's found across, in all vertebrates and in a really similar form. And I'll talk a bit in a minute about what it does in the brain and about the fact that it might have then ancient roles in the brains of common ancestors, because it seems to be doing at least some similar things in brains of different species. But it's not a brain-specific gene. FOXP2 likes to moonlight. It's also switched on in parts of the developing lung, in the intestines, and in the heart in early development. So, is that all right? And this might be some surprise to you, but this is not really surprising for people who study developmental biology. So, like other regulatory genes, FOXP2 does different jobs depending on the different factors that are around in the cell, right? So, as I said before, it acts in a kind of complex. And the importance here is that, again, you have a combinatorial system, if you like, of proteins interacting. And this is how developmental biology and also Evo-Devo kind of works, which is you don't have a new gene arising for every single new function in the cell. You recombine different, in the body, you recombine different combinations to do different things. So FOXP2 is doing some stuff in lung, guts, and hearts in early development, and then it's doing some other stuff in the brain. And we can get back to why the disorder seems to predominantly affect the brain a bit later. So what we think is that FOXP2 provides this kind of window into the biology of speech and language. We don't think of it as a gene for language or a gene specific to language. But what we think is, because we know that when it's disturbed, it interferes with the brain circuitry that we use when we're using language, that then it's an interesting entry point into those pathways and into the genetic pathways. So we get back to this slide that's one of my favorite slides, which shows you this big gap between the DNA level and the speech and language level. And here what we want to be able to do is then study these different levels to understand how FOXP2 works at these different levels. And I'm only going to focus today, in my remaining time, on two different sort of stories. So the first one is to say, well, we know FOXP2 is this transcription factor protein that switches on and off other genes in the cell. So what can we say at this sort of cellular and individual neuron level by studying FOXP2? And one thing that we can do very easily, or, well, not so easily actually, the people who work on this will tell you it's actually quite hard, but one thing that's at least easy to conceptualize is that we could find out what are the downstream targets that FOXP2 is regulating, what are the things it's switching on or off, try and pull out this network. One of the first genes that we managed to find through this kind of route was a particularly interesting one already, because it was already implicated in brain development. And this is a gene with a horrible name of contactin-associated protein-like 2, and we tend to actually call it CatNap2 for short. And what this protein does, so FOXP2, I should say, FOXP2 is on chromosome 7 here. This gene is also on chromosome 7. That's a coincidence. FOXP2 can regulate genes anywhere in the genome. This is one of the ones it happens to stick to. And what FOXP2 does is it sticks to this gene and reduces its expression, and I'll show you that in a second. And the protein that this makes likes to sit in neurons, and it likes to sit in the membranes of neurons, in the axons, right? These growths that come out from the main sort of transmissible outgrowths from CNS cells. And it clusters potassium channels at the nodes of Ranvier between myelin sheath. So this protein is able to affect nerve conductance. But it also is important for the early wiring up of the nervous system. And there's lots and lots of work that's been done on that. So, for example, it's known to cluster in this initial segment of the axon and mediate interactions with interneurons, for example. So there's loads and loads of work on this gene already in this protein. It's very interesting. But the idea that FOXP2 might regulate it was a very interesting one to us. So this is the gene. It's very, very big. It has lots of kind of interruptions in it, which many genes do, so it's a broken gene. It spans about 2 million bases of DNA, so it's a very, very annoying gene to have to study, but you can't choose which genes you want to study sometimes. So FOXP2 sits in a regulatory element near the beginning of CatNap2. And what we found in cells is that if you upregulate levels of FOXP2, you always downregulate levels of this CatNap2 gene. And this has been shown by multiple labs now. And these are slices of brain from mid-gestation fetuses. And this work was done by Dan Geschwin's group in UCLA. And what you can see here is this is the sort of layered structure of the cortex in a developing embryo. Remember that you have 6-layered cortex in mammals. And what you tend to find is FOXP2 is highly expressed in the deeper layers of the cortex, where you can see this yellow labels here, whereas CatNap2, which is labelled yellow here, tends to be in much more superficial layers of the cortex in this early development. So there's an inverse relationship between FOXP2 being expressed highly in certain levels and CatNap2 being expressed at low levels. Now, this doesn't prove that FOXP2 is repressing CatNap2 in the brain. All it does is goes in line with this kind of more direct evidence. But it's a nice kind of in vivo correlation. So we then were looking at families with Tony Monaco who had not these kind of simple forms of genetic language impairment as you see in the KE family, but with more common forms which often don't involve this severe speech dyspraxia. And what we found we were studied 184 families, and we have a number of measures of their language performance. And you find that if you span across this gene, there's a section of variants around this part of the gene which have a sort of quantitative effect on how well you perform language. So what this means is it's not like an all or nothing effect or a dramatic effect like in FOXP2. It's just one risk factor which seems to contribute to a sort of a subtle but significant reduction in your linguistic abilities. And this was seen in kids with language impairment who didn't have autism, but it's also the same effect has been shown in kids who are autistic and who also have language impairment, or they have delayed language as well. So Dan Geschwin's team showed that these same variants are associated with age at first word in autistic children, which is a very sort of simple measure, but the effect was sort of consistent. And we've also more recently, in collaboration with Andy Whitehouse in Australia, shown in a big sample that these variants, if carried in the general population, also seem to relate to early language performance. So this is an interesting finding which links this gene in some way to language impairments, but also CatNap2 has been implicated in things like epilepsy and schizophrenia as well. So this seems to be a gene that, depending on what other things are around, it predisposes you to various types of different neurodevelopmental disorders. And I should say that now, since this study, there's various different other parts of this pathway that we've been studying and trying to link to language impairments. And so an example would be, there's a gene called FOXP1 that is less well known, but it's becoming quite interesting because it interacts with FOXP2. So the final thing I want to talk about in relation to this kind of slide is the neural circuitry level. So we know that FOXP2 makes a regulatory factor that switches on and off genes in neurons. But what we really want to know is how that affects the behavior of neural circuitry, and then how that impacts on the brain. So one of the first things we can ask is, where is FOXP2 itself switched on in the brain? And this is a very simple kind of schematic of what happens if you slice through a human brain, which I don't do every day. But and of course, here's the 6-layered cortex. And what I should say about FOXP2 is it's not expressed in every structure in the brain. It's not ubiquitously expressed everywhere, like some genes are. Some genes have more selective expression patterns. And as I showed you before, FOXP2 tends to be expressed mainly in the deeper layers of the cortex, mainly layer 6, a little bit of layer 5. And it tends to be, so far as we can tell, it's quite widely expressed in the cortex, but in this deeper layer. It's also expressed in some other interesting structures. So one of the highest expressing structures is the striatum in the basal ganglia. And it's also, Cedric will be pleased to hear, it's also expressed in the thalamus. So that's another site where it's expressed, and also in the cerebellum. Now, where it is expressed in the brain, it seems to show selectivity in which type of neuronal subpopulations it labels. So, for example, in the caudate nucleus, it's very highly expressed in medium spiny neurons, particularly in D1 medium spiny neurons. In the cerebellum, there's many different cell types in the cerebellum. And in the cerebellum, you see it expressed highly in Purkinje cells, which is a major cell, one of the major input, output cell types of the cerebellum. So what you see then is kind of circuitry. So you can look at these distributed circuits, which FOXP2 seems to be labelled. So it's sort of halfway between highly selective and being distributed all over the brain. Now, of course, what we want to do then is understand what it does in those circuits. And the Institute of Child Health team, led by Farron, they've done a lot of work with just neuroimaging in the family. And interestingly, before we even found the gene, there was evidence that the family have a subtle reduction in the size of the caudate nucleus and some slight increases in putamen, and also some cerebellar. Again, these are kind of subtle effects that you see on statistical analysis of MRI. And also they've seen functional effects in the striatum and in the cortex. So at least there's some convergence. But what we really want to do is look in the neural circuitry. And there we have a problem because we can't take the brains out from the kids in the KE family or these other cases. At least the parents get very upset when you suggest it. So we then need to move to a different species. So everything, or I think everything I've talked about in relation to FOXP2 so far has looked very much at humans. But I have said that it's well conserved. So this is the favourite organism that we like to study as geneticists when we want to study mammalian genetics, because it's a very, very easy to manipulate beast. So we can change the genetic material quite easily, and it's kind of easy to breed and all this kind of stuff. So does this mouse have a speech and language disorder? Well, actually, there is a very serious side to this. Because what is FOXP2 doing in a mouse? Is it even there at all? Well, yes, here's the human version of FOXP2, and here's the mouse version of FOXP2. And actually, if you stretch it out, you see the mouse is one amino acid shorter, and that's because in this big stretch of polyglutamines, it's missing one, which doesn't seem to have much of a functional effect. And then there are just these 3 amino acid changes. So mouse FOXP2 differs from the human protein by only 3 substitutions, these 3 substitutions in this shorter stretch. So this is one of the most similar of genes in mouse genome and human genome. And if you look at where it's expressed, you see very similar. There might be some differences that we can't detect at this point, but the basal pattern is the same. So it's switched on in similar regions in the rodent and human brain, deep layers of the cortex, medium spiny neurons in the basal ganglia, dorsal thalamus, Purkinje cells in the cerebellum. These are circuits that have been implicated by many studies in mice and other species in multimodal sensory processing, sensory motor integration, and motor skill learning. So we've made a variety of mice with different things going on in FOXP2, but this perhaps is one of the more interesting ones, because here we make exactly the same mutation as in the KE family. So it's got a slightly different number, R552H instead of R553H, and that's because of this shortened Q, which just makes Q stretch, which just makes the numbering system different. But basically, it's exactly the same change. And what we can say is if you have so we can now make mice that have no FOXP2. So if we make mice that have 2 damaged copies of FOXP2, we've never seen this in humans, because all the humans that we found always have 1 damaged copy and 1 normal copy. What you see, actually, well, you can't see it so well here, is you can look in the brains and look at the genes that FOXP2 regulates. And what we found early on by doing the same kind of work that we did to find this CatNab2 gene is that many of the genes that FOXP2 seemed to be regulating were important for the outgrowths of neurites and axons, and also for the plasticity of synapses. So we went in closer to have a look at mice that have no have no FOXP2 at all, right? And if you have no FOXP2, you're basically a dead mouse, actually. They live for about 3 weeks after they're born, and they seem to have very subtle lung deficits, which may explain why they die. One of the interesting things we can do with these mice is we can take neural stem cells out and we can grow them in culture, and we can ask what happens to these dendrites and to neurite outgrowth. And here, okay, I'm getting towards the end. And here what you can see is that FOXP2 is important for neural connectivity because you have reduced neurite outgrowth. So you can measure the number of processes that come out from a neuron. So in this case, you can see we have 4 different outgrowths. And we found that actually mice with no working FOXP2 have a normal number of processes. But you can also look at the branching of these neurites. And here what we found was quite a significant reduction in the neurite branching. And also we can measure the total length. And the total length is also much shorter. So this means that FOXP2 has a very basic function which is important in early development in neurite outgrowth. Now if you have mice that have one damaged copy of FOXP2 and one normal copy of FOXP2, a bit like the KE family and all the other humans that we found, they're absolutely fine. Okay? They live full, long, healthy lives. They don't have any gross motor problems. They don't have gross lung problems. They're absolutely fine. And what I don't have time to go into is just to say that the interesting thing that we did find is that mice that carry one damaged copy of FOXP2 show normal baseline motor behavior, but significant deficits in learning and motor skills. And actually not on orofacial specific tasks, but on these kind of running wheels and some rotor rod as well. And if you're interested also in, there's a paper that just came out last year in molecular psychiatry where we looked at the neurons in the medium spiny neurons in the striatum in vivo, how they were firing during the learning of these tasks. And we found that there were differences in the neurons and the way that they fire. So, and this is the kind of stuff we can never do with well, it's hard to do it in humans, but it maybe suggests new things that one can do in humans. Anyway, so I want to finish off. You might be saying, well, I said in the last sort of 3 minutes that I have, I've said nothing about human evolution, right, really. And I think this is a key point that we can do a lot of analysis and learn a lot about FOXP2 without having to worry about this question of what makes us human and what... but at least it tells us something very interesting at this level. But it is interesting to ask, how has FOXP2 changed in recent evolution, and has that got any link to human development? And this is a question that we asked with Svante Pääbo and Wolfie Enard in Leipzig. And what's interesting here is here's this schema that, remember I said it's very highly conserved, this gene. It hasn't changed much in evolution. But what's clear is that the changes that have happened on the human lineage have happened very recently. So they've happened, these 2 amino acid changes here and here happened after we split from the chimpanzee. There's some question about the dating on this, which I'll get back to in a second. So when it comes to FOXP2 protein, the chimpanzee is more similar to a mouse than to a human for this FOXP2 protein. And this kind of looked interesting. I think it maybe has been... I think maybe it's, you can take these kind of things too far. Hold on. But one thing I really want to stress, because I didn't realize that people make this mistake, but they make it very often, is that these changes, these evolutionary changes, sometimes people call them mutations, they're evolutionary changes. They are different from the changes that cause the disorder in the family, in the KE family, in other cases. So these down at the bottom are the mutations that are seen in the disorder, and these are the changes that are seen that distinguish a human version of FOXP2 from a chimpanzee version of FOXP2. The other point that's been much debated is when actually these changes occurred, and originally it was thought based on looking at modern species and sort of modern humans and extrapolating back based on variation at the locus. It was originally thought that these changes were very recent and had happened within the past 200,000 years. It's now pretty clear that Neanderthals carried those same amino acid substitutions, which means that the date must have predated or most likely explanation is the date predates the Neanderthal split. I don't think it tells you anything about whether or not Neanderthals had language, but I think it just helps us to get a finer tune on when these changes happened. So they may have been deeper in history. But the point here is that there's accelerated change of FOXP2 on the human lineage after we split from the chimpanzee. This doesn't mean that FOXP2 was the driving force behind evolution of spoken language, but it could be interesting because
We know that it has these ancient neural circuits, roles in neural circuits of the brain that are conserved across across vertebrates, and it may have been tweaking of these kind of circuits that and all we know at the moment is that they were important at least for motor skill learning and maybe some other functions as well. So it's just one piece of a complex jigsaw. And I want to finish by saying this, which is that one of the nice things here in terms of evolutionary questions is that we can test. So we have a hypothesis that we can test, and the hypothesis is, did these changes to FOXP2 actually affect the FOXP2 function of FOXP2 protein in any way? If they didn't, then they're not going to be very interesting amino acid changes to study. Ideally, we'd like to stick these into a species that's similar enough to us that they've got a similar background, and this is maybe illustrates one of the problems with that, but maybe not. So instead of doing that study, what Wolfe-Enard did in Leipzig was to insert them in the mouse, and this is not to create a speaking mouse, but rather to empirically study whether these amino acid changes can affect the way that the brain functions in this in vivo system. What is interesting is that when you put these changes into a mouse version, you don't see the same changes that you see in the mutations that cause disorder. So it's not disrupting the gene in this basic way. So, for example, with these mutations, we see a disruption in the synaptic plasticity in the striatum. When Wolfe put these changes into mice, he sees a sort of increased synaptic plasticity in the same cells that we see reduced activity. So that's at least interesting. So I'll finish there by saying missense and nonsense mutations in FOXP2 cause this severe speech and language disorder in humans. It's a regulatory gene. It has these downstream targets and also interactors, and we think that these offer entry points into the neurogenetic pathways that are important for speech and language, but it's not a gene for speech or a gene for language. We find FOXP2 in many species, including mice, and there's a nice songbird story that we can talk about later. And we've got lots of evidence now that it's important for neural plasticity of the brain circuits in which it's switched on and off. And this, to me, suggests that our unique capacity for complex spoken language is built on evolutionarily ancient systems. And there I will finish. Thank you for your attention. These are most of my FOXP2 collaborators. It was a big team effort. And this is my old lab in Oxford, who are now relocating to Nijmegen, some of them. And I will stop there. Thank you.
Thank you. Michael?
Thanks very much. The question that I would ask is there any research underway to try and look at differential expression within cerebellum for regions involved with orofractal, orofacial, rather than limb praxis? Because you said that KE does not have limb apraxia, so that seems something that we really need to understand better.
Yeah, so one of the questions about the praxis is, or the deficits, is this the question of specificity in terms of the deficit, whether it really is specific to speech and language, or whether it's just that if you gave them a really challenging motor task with their hands, would they also show deficits? And Fariner has been looking at that. There's not much data published on it yet, which is a shame. But I think it's a key question to ask as to whether it's just that speech is such a demanding, this kind of fine, rapid, coordinated sequencing is so demanding that maybe they do have other deficits that only show up in the context of speech. Whether it's related to the cerebellum or the striatum, we don't know actually, because they're these two circuits where FOXP2 is expressed. So, but yes, that's a very good question.
We have only 5 minutes, but where? On the back. No. Yes.
Thank you for summarizing complex material and presented in a clear way. You were very careful to say that FOXP2, you said, is not the gene for language. You said it many times. However, during the talk, you also used in an interchangeable speech and language, spoken language, and so on.
Yeah.
My question is, since there are so many dozens of languages on Earth that are non-phonated today, What is your impression of what do we really learn about language proper, and what do we learn about vocalization and speech?
Yeah.
And can you say a little bit more about that? Thank you.
I think that's a really tough question to pull apart, because these kids grow up with the speech problems early on, and pulling them apart, the cause and effect in relation to their language. I mean, I don't think it's. I think that, so. There's a question. I think Massimo covered this the other day, which is it pure? Is language purely motor, right? And he would say no, because you can have severe motor deficit and still have intact language. So the question is, in the case of the KE family and these other cases, are the linguistic deficits completely just a secondary cause of a basic speech or motor deficit, or is it that the circuitry is pleiotropic and the same circuits are implicated, same basal ganglia circuits, for example, are implicated in aspects of these grammatical aspects and the speech aspects? And I don't think we can answer that question at the moment, but what would be good would be if we see mutations in other. I mean, I would love to find out what, like, if somebody had a click speaker, and whether how they would be affected by FOXP2 mutations, and then other languages that have so I think these kind of questions need to be answered. But I appreciate I'm a bit fluffy about the sort of speech and language things, because I don't really know which is the core deficit.
Excuse me. One of the things that has interested me about FOXP2 is its incredible variability in bands. As an evolutionary question, bats, of course, use remarkable vocalization. And yet it's incredibly variable compared to almost all the other mammalian groups, which are quite tightly clustered. Is there anything that you can say about that variability? Does it tell you anything about its evolution and its growth?
Well, one problem with the variability in bats, the story about the variability in bats, which is a really interesting finding, is knowing how that lines up with that, because only a few genes have been studied in bats. So knowing how that lines up with variability generally, background variability in other genes, and whether this really stands out as much as it might do. But what's interesting is that the variability doesn't relate to vocal learning, it seems, but it looks more like it relates to echolocation, and which is interesting because this might fit in with ideas of integrating sensory motor learning and sensory motor information, which is, of course, important in relation to speech as well. And there's a study that should be coming out soon on bats that sort of supports that idea. So.
Well, thank you. Wonderful talk, really wonderful talk. I have two questions. Some affected members of the KE family, they have learned British Sign Language, and I'm curious to see how good they are at acquiring British Sign Language. And the second, if you can tell us what about the latest in the activation of FOXP2 in some birds, conference sharks and others have done wonderful work, activation of FOXP2 in songbirds. What are the latest about that?
Okay, so the first question. One kid in the KE family has learned sign language, I think. But again, there's no anecdotal evidence. I think that would be really nice. That's another way of separating out the speech. Well, maybe not, because again, it's motor. But I think that's an unanswered question still, of the issues of would a kid with FOXP2 mutations show similar deficits in sign language. And the songbird story. So if people aren't familiar with it, the basic story from a couple of years back is that when a constant shark knocks down FOXP2 in area X of the songbird, which is part of the vocal learning circuitry, that the birds who had this sort of half dosage of FOXP2 in that striatal nucleus, they showed impaired learning of songs, and it looked a bit like a verbal dyspraxia phenotype, in that there is reduced accuracy of copying and emission and all sorts of things like that. So it's a nice, the songbird model is very, very nice in this respect.
Thank you very much, Simon.